I want to transition a little bit, talk a little bit about your research. It seems like from your kind of CV background page you sent me, you've done a lot of different things. I will probably have to stop you at various points and ask explanations on various pieces of jargon as I, try to work my way through what you've done. So can you start with like what you worked on as your dissertation for your PhD, and we'll kind of try to move forward from there or hit the high points, I guess.
Sure. So I will preface this by saying I did wind up doing my PhD, postdoc, and now my current position all at Penn State University. That was not my original plan. It is discouraged, especially in life sciences and some other fields as well, to stay in the same spot. And it's considered that you were, not really broadening your horizons and your ability to contribute, intellectually could decrease, the longer you're staying at a certain place. But, I made the decision to stay for personal reasons. I met my future husband as I was finishing up my PhD. So that's why I stayed and I'm fine with my choices in that respect. So when I was did my PhD, I studied muscle development in fruit flies. And the first thing that a lot of people are like, oh, fruit flies have muscles, but they fly and they walk. So yeah. And we specifically studied embryonic muscle development so that the muscles that would form when it's a developing embryo and that the larva would use to crawl around on, the meat or whatever they're going to eat. And I studied a gene called Nautilus, and what we realized that gene does, there's several, they call paralogs, and what that means is related genes in like vertebrates, and they're like muscle regulatory factors. And the gene that we studied in Drosophila was responsible for differentiation of muscle precursors. So when muscles are first forming, they don't look like a muscle. They don't look like a striated muscle, right? It just looks like a round cell that might express some muscle proteins like myosin and actin. But when you're actually differentiating or forming into the more mature muscle type, what are the genes responsible for that process? And we found that this gene Nautilus that I worked on was responsible for a subset of muscle differentiation in the embryo. And we use a variety of different approaches, a lot of genetics and molecular biology. I did a lot of microscopy during that time. So I really felt like it was a really good experience. I felt like I learned a lot. And I came out of there, though, and kind of wanted to switch things up a little bit. And like I said, I stayed here at Penn State, so I wanted to broaden my knowledge and my experience base as much as possible. So I did switch fields. Some people will actually stay in the same field that they get their PhD in. And for me, I decided to switch and did GABA receptors in mice. And GABA receptors are responsible for the majority of inhibitory neurotransmission in the central nervous system. So to kind of, calm things down, right?
Mm-hmm.
And so that again was, I did a lot of work with mouse neurons, primary mouse neurons that we had to dissect out of these tiny little 14-day embryos, which I felt very bad about. But the worst thing actually was when I was doing these mouse dissections is doing these with little forceps, and I had to do it under a dissecting scope. And every time we did dissections, I could not drink any caffeine that morning. That was probably the worst thing about it, right? I was like, I really want a cup of coffee, but it was so tiny that you really did not have the control, to really get in there and do these
So
dissections.
your dexterity would be affected by the caffeine enough.
Yeah.
Okay.
And I mean, because not any noticeable. It's not like I would notice it if I was, taking notes with a pencil or doing anything else. But when you're actually trying to, dissect really, really small things under a microscope, it was just enough that you lost a little bit of that control. Yeah. So that was, but so I did that for a couple of years, and I wound up studying this one. So these GABA receptors, the gamma subunit. So GABA receptors are made up of a number of different proteins that together form the receptor. And one of those proteins is this gamma subunit. And I worked on trying to find various proteins that might interact with this gamma subunit to try and control, to try and learn more about how these GABA receptors cluster in synapses. So what you, in general, right, so receptors, neuron receptors will be at synapses where you have an axon and then you have your dendrite, and your receptors will cluster together underneath the axon to receive input. And what we were trying to figure out is what are the mechanisms behind clustering of these GABA receptors at synapses. And so I did some screens to look for interacting proteins, and I discovered the first mammalian palmitoyltransferase. And what that means is it is an enzyme that attaches a palmitoyl group, which is a fatty acid, to this gamma subunit, which would then affect its clustering. It would allow it affect how the receptor gets to the cell surface and gets recycled back and so forth. So that's sort of what I worked on as a postdoc. So I kind of, I did a couple, already I'm switching model systems. I worked with fruit flies and I worked with mice. And I enjoyed both those things. And after that, I kind of felt like I needed a break from academia. And I thought, I need to kind of get out of here and see what else is out there. Unfortunately, here in State College, Pennsylvania, I mean, Penn State is the largest employer of science people. There are a few companies around, but there's really not a lot of options as if there would be, in a bigger city where, there might be pharmaceutical companies or biotech, and we just don't really have that here. But I did work for a small company who did mitochondrial DNA forensics. So this is back 2004, and I'm trying to remember when the human genome was first sequenced. I want to say 2001.
That's what I wanted to say offhand, but I would trust you more than me on that.
Say that around that, a little before that, but it really wasn't. Nowadays, you can do whole genome sequencing pretty fast. In fact, that's what I do for a living now. But it's DNA sequencing. But back then it was harder to sequence DNA from blood and so forth. But the other thing too is that mitochondria, so red blood cells, do not have mitochondria. Well, no, they don't have nuclei. So you have, if you wanted to isolate DNA from red blood cells, you could get mitochondrial DNA, but you're not going to get nuclei DNA, so genomic DNA. So, but anyway, we would, so a lot of what we would do would be to get like samples of bone and hair in, especially a lot of hair samples. And a lot of it was cold cases where we would isolate mitochondrial DNA from old hair samples. And a lot of it was trying to more like exclude people as being, exclude people from, committing the crime, right? So were they convicted and based on some evidence, but there was not any DNA evidence, and there might have been some old hair samples or blood samples or something from the case, and a lot of times we would test those and look to see whether there was a match to the person who was being accused of the crime. So it was interesting. I learned a lot about quality control at that point, and really, taking, not that I was not careful as a scientist, but when you're thinking that you might have somebody's not actual survival life on the line, but their livelihood in terms of whether or not this person is in jail or not, right? You realize, you take a lot of responsibility for that and be like, wow, I'm really could potentially really affect somebody's life. And I think it really kind of stepped up my awareness in terms of, sample handling and quality control and things like that. So I think that actually I made for a good transition to my current position,
Mhm.
which I'll go into in a minute. But I actually wound up, after I left there, I took a couple years off from that. I sort of stayed at home and worked part-time when I had my son in 2005. And I never really left science. I taught a couple of classes in the evening, a couple of molecular biology classes, and I did some freelance work for some publishing companies where I wrote some test questions for online material, study material, and things like that. Before then, I got my current position, so as an associate research professor, and we study blood cell development. And mostly I do a lot of DNA sequencing, which is really kind of fun.
So I want to back up a little bit. You were
Oh,
talking about no, you're fine. I was just like trying to keep notes and try to like remember. Okay, I want to ask about this and that. There was something about Nautilus I wanted to ask, but I just wrote that down and then I lost my question.
sorry.
No. Well, maybe I'll think of it. So I was curious with you're taking samples. You're saying you're taking or doing like sequences of samples from cold cases. This is just a pure personal curiosity, but like, is there a shelf life on that kind of thing? I mean, I would think that like say you had a bone fragment for some reason. Well, like the bone fragment in storage is going to be fine. Like it's not going to decompose. But I just wondered if at the DNA level, if like there's like unraveling over time, or if like this integrity of the sample can be relied on over a variable amount of time.
Sure, that's a really good question, and there is some level of degradation of nucleic acids over time, and but some of the bone fragments we worked on, were decades old, and it kind of sometimes you would get some high quality mitochondrial DNA from that, and sometimes you wouldn't, and excuse me, we would. In order to sequence some of that mitochondrial DNA, we would amplify using PCR to amplify different portions of the mitochondrial genome. And we would use several different areas so that you're not just relying on one portion of the mitochondrial genome being intact. Okay, so you may get, let's say, and I don't recall, honestly, to be truthful, the exact number of ones we did, but let's say for each sample you try, 4 sites, and you may get amplification of all 4, or you might get amplification of just 1 or 2, depending on the age of the sample. And then you can, but what we always do is compare it to a known, right? So if you had sequence from your unknown, and you got some sequence from that, and then you compare it to a known individual, and does that match? So at one point, depending on the level of sites we were able to amplify, or sections we could amplify, and the amount of match, a lot of times it was more like you would exclude somebody more than say it's definitely this person, right? Mm-hmm. So it was more of a system of exclusion, because mitochondria are transmitted from mother to son and mother to daughter, right? So you're always getting your mother's mitochondrial DNA. So you would have the same mitochondrial sequence as your mother. So you really couldn't say it was you versus her who committed a crime, right? But it could exclude you from being a person from a different mother.
Right. Oh, well, it's essentially just another method to move forward. Whether you're trying to include or exclude somebody, you're approaching the problem from two different sides essentially, which depending on what you have to work with, like you mentioned, just like sometimes it's just pieces instead of the entire sequence. You do what you can and what makes sense versus what you necessarily want to do, which is like, no, this is absolutely we have the whole thing, it matches perfectly. It's like, no, you just work with what you can.
And there is also no mitochondria. So unless you have the hair shaft, the actual part that's stuck in the head, then you don't have any genomic DNA, right? So when you're actually just so a lot of times with hair samples too, you're working with more of a little piece of a hair sample, and that does have mitochondria, but again, unless it has the shaft, then you can't isolate genomic DNA. So there are advantages and usefulnesses to still doing some mitochondrial DNA forensics. It's just that with the advantage of advance of genome technologies and sequencing nowadays, if you have genomic material, it would be much more clear of whether there's an actual match or not.
Mm-hmm. So, I mean, it seems like no matter what project or research project you're working on, like you seem to be focused on like the gene sequences and trying to figure out, what genes play a role in whatever it is. Like, please correct me because I'm going to get this wrong.
Sure.
Like when you're talking about nautilus and the development of muscles in the fly embryos, correct? You're trying to figure out what do I want to say? What mechanism or what gene? Should look for the word. Um, what thing? I'll just say thing, and you can give me the actual word here in a minute. Was telling the muscle to create this particular muscle, right?
Right. Actually, that's a great explanation, and that it's really about gene regulation and how genes are turned on and turned off, and what are the processes involved in how genes are turned on and turned off, and what networks might be involved or other processes. And I think a lot of those will affect cell fate choices. Do we want to become we work on blood cell development now, and when we start with a stem cell, is it going to eventually become a white blood cell, or is it going to become a red blood cell? And we look at how that cell fate decisions are made, and by learning more about those processes in a normal cell, and again how genes are turned on and genes are turned off, and all those regulatory mechanisms, that can provide some insight into disease processes. So you really got to kind of learn what's normal and how things work normally, and then you can ask those same questions by looking in some sort of disease state, and learn more about that, right? Which down the road, your ultimate goal really is to improve, human health and performance and so forth.
And that's kind of what the Vision Project's about, right? So trying to figure out. So I tried to write a summary. You provided much more detail, but basically looking at genetic traits to determine like susceptibility to disease or resistance to a particular treatment.
Exactly. So Vision, which is sort of a bit of a acronym, stands for Validated Systematic Integration of Hematopoietic Epigenomes, which is a big mouthful. Really, what it really involves is that right now we as a scientific community who are involved in a lot of gene regulation and DNA sequencing and so forth, means the amount of data that's being generated is tremendous. And data generation is no longer the limiting factor. Back when DNA sequencing first became more widely available, it was, this big rush. Let's sequence everything, sequence it all. And, and everyone was super excited, but there was a cost factor in that it was very, very expensive to, both in reagents and equipment and processing and all that. It's very expensive and time consuming, but data generation is no longer the limiting factor. And it's more now is how do we use all that information to extract what's relevant and make hypotheses and predictions and then test them. Right? So it's great to generate data, but unless you have ways to analyze it and pull out what's important, it's not that useful, right?